Okay, so good evening. We are going to have an exam today, like I had already mentioned over email. So, just to brush up, the topics that we are covering are largely around the basic strategies for template amplification, sequencing by synthesis, sequencing by ligation, and it's a pretty simple test. I've kept more number of questions this time. There are 45 questions, no negative marking. And of course, it's not that very sort of formal sort of an exam, so don't worry. You'll get half an hour to be done with it because it's quite simple. So I'll just wait like for 2, 3 more minutes if somebody else just in case joins. But in the meantime, I will share the link with you so you can access it and you can answer the questions and you'll get your response after completion of the test. So I think 2 or 3 people appeared for it last time, so we'll wait out for that duration. Um, and following that, if there's anything else that I'll discuss the paper, of course, with you. And if there's anything else that you wish to be clarified, we'll discuss that. And next, see 3D chromatin architecture, what sort of conclusions you can draw from such techniques, what is their utility, all of that is what we'll cover from that point onwards. So I will share the link in. Let me see whether it is accessible or not. Just a minute. My system is a bit slow today. Yeah,
Are you sending the link on mail?
yeah, it's just I'm sending the link over. No, I'm sending it on Teams only, so that you can start it right away. And if whoever wants to attempt it later on, I'll share it over email, and they can check it out from there. So uh, it's a little slow, but I think it should switch off the camera. I think that's slowing down. Yeah, let me know if you can see it. I think it should be accessible. Hello?
Yes, ma'am.
Yeah,
I'm
did
sorry.
you get the link?
Not yet, ma'am. I think
I sent it in the meeting chat. Can you check that once?
yes, ma'am.
It's a Google form link.
Ma'am, it is not like I'm not finding the link.
Okay, then I'll send it over email. Just give me a minute. Yeah, check your mail now. It should be accessible.
Just a minute.
Yeah.
Yes, ma'am. Now it is. The link is
Okay.
sent.
So by 7:15, let's try and wrap up because it's not a very lengthy test. So then you can we can resume our discussion. Okay. So good luck. Please let me know if there's any confusion, okay?
Okay, ma'am. Ma'am, in the 44th question, I'm unable to like the second option is I don't know. I'm not able to tick that option. Ma'am, am I audible?
Yeah, hi. In the 44th question, exome capture, that question.
Yes, ma'am. The hybridization probe is like I'm clicking it, but it is not.
Um, like from my end, it is accessible. You can,
Like, ma'am, all the
if you,
other three options are like I'm when I'm clicking,
uh-huh.
the cursor is like it is showing, but on hybridization probe, it is not like even when I am clicking the option, the cursor is not showing there. I don't know why.
Okay. Like from my end, it is visible, but it's okay, no problem. You can move on to the next question. Like it shouldn't happen because, yeah, from my end I can see it. But okay, no problem. You can do the next one.
Okay, okay.
Okay, so you're done, and I think you submitted it also.
Yes, ma'am.
Okay. Okay, I'll discuss the answer and then I'll release the score. Um, did you skip a few questions? Acha, no, you got
No,
42.
ma'am.
One of them, I think, is that exome capture question that was creating some trouble.
Yes, ma'am. I think
Okay, so
40, 44th
okay,
question.
okay, no problem. Uh, okay, and I think I put the wrong answer for one of them. Just give me a minute. Okay, so you got just one wrong. The rest, everything is fine. So very well done. I'll share my screen and discuss it with you as well. Just a minute. Okay, so the PPT should also be open. Just a second. Yep, the PPT is also open here, so we can go back for reference if needed. Okay, so the first question was about second generation sequencing. Maybe I'll open the PDF side by side, or sorry, system is not working well today. Yep. Okay. So on the right, I've given the syllabus. that we are following. On the left, we have the questions from today. So, first question was about second generation sequencing and what it is usually characterized by. So, single molecule, massively parallel sequencing, or Sanger chain termination, protein sequencing. Of course, the last two options are naturally not correct, but between single molecule and massively parallel sequencing, the correct answer is massively parallel because single molecule sequencing goes a step further that is not covered under second generation sequencing process. So this is, I think, what we had started off with initially. So this is where we initially discussed second generation sequencing, as you can see on the right. And we discussed the different types and what exactly is included, or what are the strategies that are included under second generation sequencing, which included the Solexa technology, the pollinator, the heliscope single molecule sequencer, etc., which are all part of it. But the overall idea is that you go for sequencing, which can be done for short like short fragments, but at the same time and eventually give you high throughput results. So the second option is correct, and you got that right. Second, in Illumina sequencing, how are nucleotides labeled? So, radioactively, fluorescently, unlabeled, Python labeled. Again, this was quite simple, and we had discussed case. Yeah. So we had hope you're in now. Okay. So we had discussed the idea of Illumina sequencing, how it works. If you go back to this slide here, you'll see the Illumina Alexa sequencing strategy. And under that, we had discussed how this process of extension and termination, and then again the addition of the next nucleotide and the consequent emission of a certain type of fluorescence, all of that is taken care of. And this entire process is facilitated by the reversible terminators that we discussed also in the previous class. So option 2 was correct. Then we moved on to what ensures one base is added in each cycle in Illumina sequencing. So what is this specificity, or what is this control maintained by? Again, by the same reversible terminators. They ensure, again, this is the I kept it simple, so they kept it in tandem. So basically, the idea is that the reversible terminators are going to do both of these things. They are going to allow us to repetitively use the same concept over and over again. Hence, the reversibility becomes useful because it is a chemically cleavable end, and you can get rid of the incorporated fluorescent dye, and then the next base is ready for addition. So uh, that is what is taken care of by the Illumina sequencing strategy. Then the bridge amplification strategy, also taken care of by Illumina. What is the surface on which it occurs? The PCR tube membrane, flow cell, agarose gel. Of course, it is the flow cell surface. That's where it takes place. Um, if you had a different template amplification strategy, for example, uh, if you remember the strategy that I discussed about the emulsion PCR based template amplification, then in that case, the correct answer would have possibly been the PCR tube, because, or any tube for that matter, where within the tube you have the micelle formation, and within each of these, not micelles, the droplet formation, and within the droplets, these individual PCR reactions, they take place that can be eventually used um, for the sequencing protocol. So in this case, the correct answer was flow cell surface, which is where the bridge formation and overall the Illumina sequencing strategy is carried out. A key limitation of sequencing by synthesis. So again, if we go to the slides on the right, so this is where we talked about sequencing by synthesis, the four possible strategies. So no, this is not right. This we talked about template amplification. sequencing by synthesis was started off here. We had 454 pyro sequencing, then we had the iron torrent strategy of sequencing, etc. So when we talk about sequencing by synthesis, where do you get where can you have a disadvantage? Low throughput, high error rate, short read length, or no need for amplification. Now, this fourth option is obviously not true. Template amplification is an important aspect of this. Low throughput also is not really a problem because massively parallel sequencing is something that is taken care of by all of these strategies. Long read sequencing is where you can potentially get low throughput because more resource investment is directed towards scaling up the not scaling up, more resources dedicated towards sequencing long continuous stretches of your DNA by nanoball generation, and therefore the throughput or the magnitude to which that strategy can work is limited, and therefore that also is not the correct option. Then comes high error rate in homopolymers or short read length. So usually it is actually short read length that can limit sequencing by synthesis because it cannot continue for an extended duration. If you, I think I don't have it in this slide, but anyway, never mind, because it carries out the sequencing process by incorporating one base at a time. Therefore, very long stretches of DNA cannot be sequenced by this strategy, and so this is its key limitation. So that's what we see here as the correct. Question 6, we were asked which step follows nucleotide incorporation in Illumina sequencing. So right after nucleotide has been incorporated, what do you get? You get the signal emission, which can be detected using your laser detectors or using your machine, whichever is able to convert that signal into a value, and then you can automatically trace back which base has gotten incorporated at that position. So signal detection is the obvious correct choice in this case, and definitely an easy question to answer. Then the term paired end sequencing, what exactly do we mean by it? Do we mean two different samples getting sequenced? Are there two different polymerases which are taking care of this? Two flow cells or sequencing both ends of a DNA fragment? So in general, this is the strategy, paired end sequencing strategy refers to two different enzymes or two different polymerases simultaneously or at a similar pace are able to carry out sequencing from both ends of a DNA fragment. However, this particular possibility of using two different polymerases can exist if you look here, while technically it doesn't happen. But in the smart bell template, because you have two ends, it is possible that there can be two different polymerases that potentially can carry out sequencing at the same time. However, it will not be recommended. It can lead to bias, incomplete efficiency, or inaccurate results that you cannot rely upon. So for any sequencing reaction, usually the preferred reaction mix has one polymerase only, which is able to catalyze the entire process. So this is the correct option, which is option B, sequencing both ends of a DNA fragment. Then you have Illumina sequencing primarily detects which of the following. So So the options here included pH change, fluorescence emission, electrical current, or radioactivity. So this pH change is detected in this strategy by iron torrent. So they actually monitored this change in H plus ion release, and that is what is monitored. Radioactivity is not the correct option. Electrical current again is not the option here. In case of iron torrent, that's what works. And so the correct option in our case for Illumina sequencing is going to be fluorescence emission by the fluorochrome, which has been tagged to your nucleotide base of interest or DNTP of interest. Then in question, I'm just rushing through this a little bit because you've already attempted and you've done very, very well. So I'm just going through the questions a little fast. Any point, any doubts, any queries, please let me know. Please interrupt me and we'll discuss it. Next, you have cluster generation. What does it exactly end up increasing? The read length, the signal intensity, error rate, or fragment size. If you remember, again, this is not something I need to show this thing, but yeah,
Yes, ma'am.
basically,
The questions were easy. You can just go through it. Like,
okay, cool. So cluster generation in this case is able to increase the signal intensity, which is why we do template amplification, and that's how we detect it. Then comes sequencing by synthesis. What is it the most useful for? Again, high throughput short reads, which is what you saw in the first previous question as well. Then sequencing, okay, the other options are obviously not going to be true. Then you have sequencing by ligation, which is used by which of the following platforms? Again, if you go here for sequencing by, I think it was previous to this ligation. Just a second. Yeah, this is where we have sequencing by ligation. So Illumina doesn't use this. We have it on the solid platform and we have it on complete genomics BGI. So the correct answer is solid sequencing. Then which enzyme is central to sequencing by ligation process? Polymerase, ligase, helicase, or reverse transcriptase? The correct option in this case, of course, is going to be ligase. But you definitely need polymerase as well for any reaction. Generally, for any other strategy, any other template amplification strategy or sequencing strategy, polymerase is going to be indispensable. But because in ligation, we use these probes where we have one or two known bases, along with that, a mixture of random bases. So the job of polymerase is already taken care of. Instead, it is ligase, which is going to decide whether or not a particular probe should be incorporated. And based on that, the reaction is completely driven. In solid sequencing, probes are single nucleotide, dye-base encoded, RNA-based, or protein-based. So here the correct option is dye-base encoded because they have two bases that are known. However, it can also vary depending on what sort of probe you prefer to choose. Sometimes you can use a couple of two or three sequences that you already know are expected to be repeated or are expected to be present, and based on that, your probe designing will also vary. Then a key advantage of this entire process of ligation-based sequencing, what does it do? It gives you quite high accuracy because two bases are already known. So any of the other protocols for sequencing will always rely on the efficiency of the reaction throughout the process. So if at any one step the enzyme or the detection system it goes wrong and it incorrectly registers the incorporation of a base, then that can be read as a mutation. However, in the sequencing by ligation process, in each step you are double checking and you are ensuring that only if the complementary arrangement of bases is present, only then will my probe get incorporated, and therefore the accuracy or the efficiency of the technique increases by a lot. Then, how does it actually detect the signal sequencing by ligation? If you look here, you can obviously see that apart from the probe, the tag or the signal that is actually that can actually be read in this case is the presence of a fluorophore or a fluorochrome towards one of the ends of the probe, and therefore fluorescence-based detection works for sequencing by ligation. pH change, electrical current, mass spec, of course, are for other processes. Then the readout in solid sequencing is which of the following base sequence, the color space, amino acid sequence, and RNA sequence. What you get actually is the color, and from the color you can trace back. Let me go back to this slide again. Yeah. So what you actually get is the base which has gotten released. Your probe sequence is already known, but that is not your readout. Your fluorescence or color is your actual readout in this case. A limitation of this process of sequencing by ligation, what is it? Is it is not very high cost, but instead it is the complex data decoding. Because while on one hand we have understood that the accuracy of the technique is higher because there is double, triple validation that at position X, a particular nucleotide is present, X plus one, the next one is present, and so on and so forth. So the stitching of this string becomes very accurate, but at the same time very laborious because you'll have a lot of repetitive data that needs constantly checked basically. So the iterations that go into this particular process are affiliated with high cost, and so that can potentially become a limitation. Then you have ligation-based sequencing reads. Are they longer than Illumina? Possibly not. RNA-based, no. Single molecule, no. The correct option is that they are usually shorter, but they have high accuracy, but at the same time, the data decoding becomes complicated. Then short read sequencing, typically what length of bases does it produce? So again, this was present in our slide, I think itself. Yeah. So typically short read sequencing gives you somewhere between 50 to 300 base pairs, whereas for long read sequencing, you'll have more than 4 megabases that can be decoded. So the correct option is 50 to 300 base pairs. Long read sequencing platforms, what exactly do they include? Illumina, Solid, PacBio, Nanopore, microarrays, etc. In this case, the long reading platform, it usually is including PacBio and Nanopore. So here are the examples we saw: Pacific Biosciences and Phoenix Genomics and Illumina are also there. But where did I choose this? Yeah, I think this was, yeah, because this was real-time long read sequencing. That's why the correct option in this case um, is PacBio and Nanopore. Whereas if you include the other strategies, which are synthetic long reading strategies, then in that case, you would also include these. The Illumina strategy and the 10x genomic strategy, but they fall under synthetic. A major advantage of long read sequencing is A, lower cost, B, better resolution of structural variants, C, faster runtime, or D, no errors. In this case, for long read sequencing, structural variant resolution would also be quite useful. Cost, of course, is more, runtime is higher, error-prone nature is higher. So the only advantage that remains is that you get way more in-depth information about structural variants, or at a much more resolved scale, and as a result, it's better. Next, short reads are best suited for which of the following? De novo assembly, structural variant detection, epigenetics, or variant calling in known genomes. So for de novo assembly, this is not a very good idea. You should preferably have long read sequences so that stitching becomes easier. Structural variant detection is not very easily usable, or not very easy to interpret by short read sequencing. Epigenetics, of course, you'll not get any idea about it from this sequencing strategy. So variant calling is what works best and is actually recommended when you want to identify any mutation, any alteration, any point mutation, or such minor changes. For that, short reads are best because you get better resolution. If, like, within the short region, you will have more number of reads supporting your data. Then long reads actually end up resolving the repetitive regions much better. I think it is, yeah, my system is again hung up. Yeah, so long reads are better when you wish to resolve more repetitive regions, because there, if you use a short read sequencing technology, then it will be very difficult to stitch the correct sequence or the order of the bases, and it would give us errors. Yeah. So as a result, repetitive regions can be better resolved by long reads. Now, despite its utility, what is a major disadvantage of long read sequencing? So it usually is affiliated with high error rate. Why? Because very long stretches of DNA are synthesized, and also they form these nanoball-like structures. So they are usually error prone. Then 25, when we talk, just give me a minute. When we talk about hybrid sequencing, what exactly does that refer to? It refers to combination of short and long reads that are both being sequenced, and that, okay, this question I did not directly pick up from any of our slides, but somewhat you can try guessing it, because among all of the others, this short and long read simultaneous sequencing or combinatorial sequencing is referred to as hybrid sequencing. Which technology produces the longest reads, whether it is Illumina, Solid, Nanopore, or microarray? The correct answer is Nanopore. Then real-time sequencing, what does it measure? DNA mass, nucleotide incorporation while it is happening, RNA transcription, and protein folding. So real-time sequencing actually monitors the incorporation of one nucleotide at a time, and in real time. So that is the correct option. Then you have pack biosequencing or specific biosequencing. What is it based on? It's based on real-time sequencing data. Uh, and yeah, it was shown here in this slide. Sorry. In this slide, so by Pacific Biosciences, the zero mode waveguide um, wells are within which are part of the flow cell, within which the real-time sequencing takes place. Next, nanopore sequencing. What does it exactly end up detecting? It detects the ionic current changes, like I just mentioned. So you sort of have like a voltameter, which is able to recognize these differences and quantify them as well. Yeah, based on which you can tell which base has gotten incorporated. Then synthetic long reads, how are they generated? Direct long sequencing, assembly of short reads, the second option is correct. So synthetic long read sequencing bypasses the cost and the labor that is associated with real-time long read sequencing, and instead what it does, it sequences shorter reads and stitches them together or assembles them together. A key advantage of real-time sequencing, what does it do? It directly is able to identify base modifications. If there are any, amplification is not needed. Short reads are not limiting it. This is the correct option in that case. If there are any base modifications at in real time or at the same time, it will be able to detect them. Then synthetic long reads, what do they improve? They improve the accuracy of assembly because long reads are necessary to carry out de novo assembly. And yeah, speed and PCR efficiency certainly cannot be matched. So, or protein detection as well. So PCR assembly accuracy can be maintained, but PCR efficiency will be limited because of such long stretches of base pairs that are getting synthesized. Uh, real time, yeah, real time sequencing platforms are typically A, Illumina only, B, PacBio and Nanopore, C, Solid only, D, microarrays. So real time sequencing platforms usually are PacBio and Nanopore. Uh, so that is what works there. This usually can get a bit confusing if you are not very familiar with the way each of these companies work. So just remember the principle that is followed by each of them. Some of it is often even hinted in the name itself. Then we have the genome referring to. only coding DNA, entire DNA content, RNA content, protein content. So genome refers to the complete DNA content. And yeah, that's what we mean here. Transcriptome naturally includes all the RNA molecules that are expressed. The epigenome, what does it consist of? You have, oh, sorry. Epigenome consists of DNA mutations, sequence, chemical modifications on DNA and histones. So DNA methylation is part of genome, transcriptome, epigenome, or proteome. So in this case, it is an epigenetic or an epigenomic modification, which means the modification is not within the base sequence, but is instead it is deciding how DNA may or may not be expressed. So it's part of the epigenome. RNA-seq is used to study the transcriptome, obviously. Epigenome, proteome, and genome can be studied by completely different technologies. Then you have the effect of alternative splicing. It affects the transcriptome here. And whole genome sequencing includes only exons, introns, entire genome, or only regulatory regions. Of course, it includes the entire genome. So this is the correct option. Then WES targets or whole exome sequencing targets the entire genome, coding regions, only introns or only promoters. So WES is meant to target only the coding regions of the genome or the exomes. It's the second, it's again stuff. Exons represent approximately 50% of the genome, 10% of the genome, 1 to 2%, or 90%. In this case, the correct option is 1 to 2%. So most of the genome actually is non-coding, and only a very, very minute fraction of the same is involved in representing coding genomes or coding genes. Then WES is useful for which of the following? Structural variants only, or protein coding mutations, RNA analysis, or epigenetics. So WES is, or the exon sequencing or exome sequencing is going to give us functionally relevant mutations, or at least coding mutations. And so that in this case is the correct option. Then just then this is the question that you were stuck on, and like I could see that it's accessible from here. Like you can see this answer is marked. I'm not sure why it wasn't coming up in the paper. But yeah, exome capture is done using hybridization probes like you were already attempting, which is the correct option. And finally, if we draw a comparison between WES and WGS, then among the two, for WES, what is more relevant is the fact that it is not very data intensive. It is not more more costly. It is more pocket friendly. But again, if your objective is completely different, if it is de novo genome assembly of an organism, then you're addressing now completely different manner. You use a long read sequencing strategy. So yeah, that was pretty much it. Let me check. Yeah, it's fine. So I think only in one question you possibly have accidentally clicked this. The transcriptome consists of proteins is what you had accidentally marked. So it should be the entire coding RNA and all of the RNA that are present inside. So yeah, that is pretty much it. So good job. Thank you for joining. And I will hopefully have one more test by the end of this course. I think I discussed it in the last class that we'll have it towards the end, like in the last or the second last class, we will just have the last test. So anything else you wish to discuss, then we'll take that up. And if not, then we'll wrap up the discussion.
No, ma'am.
No. Okay, so in the next class, I will begin the discussion on ATAC-seq and 3D chromatin architecture, ATAC-seq, and a little bit of introduction into high-C. So that will be on Friday, on 20th, on Friday, we'll have that discussion. And yeah, that is pretty much it. Then thank you so much for joining and also for doing a very good job. I know it was an easy exam, but I didn't want to get into too much that will not be very useful for you eventually. This knowledge is good enough for any preliminary examination that you wish to clear. But yeah, if you want to go into more depth, then opportunities are endless. So, anyway, thank you so much. I'll end today's class and I'll see you again on Friday. So, thank you. Have a good night.
Thank you, ma'am.